Today I will briefly be going over an antibody identification using the gel method. A tech should be both familiar with the tube and the gel method because each test has its ups and downs. However, the gel method is known for being more sensitive than the tube method. To save some time for this video, I have already completed the type and screen for this patient. As you can see, screen 1 and screen 2 have both came back positive for an antibody. Now, what I would do is I would check to see if I had enough specimen left to continue on with the antibody identification. Most facilities will ask for 2 pink top or 2 blood banking tubes to do a full antibody identification. Since I am doing a video on a positive antibody, I am using these 2 cells for competency testing. They are already screened for 1 antibody. That way I can show you the positive results later on. The red cells that you see are going to be used as the patient's red blood cells, and the clear cells are going to be used as plasma. Like the screen cells, we will mostly be using the plasma portion during the antibody identification. For this antibody identification, I used a 11-cell panel. Also, it is important to note the expiration date and the lot number of the panel you use. That way, you can always check to see if you are using the correct anagram while you are doing your antibody identification. Prior to your antibody identification, you will want to gently mix each of the panel cells to ensure that the red cells have been evenly distributed throughout the bottle. You will also want to give the panel adequate time to come to room temperature. These are the IgG cards that are used for antibody identification. As you can see, there is a solid down at the base of the well, and there should be a thin layer of liquid on top of that. They are covered in tinfoil, which you pull back to expose the wells. As you can see, each card contains 8 wells. You will want to ensure that you have enough wells for your panel, plus an extra well for the auto control that you run on each patient. As always, you will want to follow your facility's procedures when labeling these cards. However, for the purpose of this video, you will want at least 2 patient identifiers to label the card. Lastly, you will also want to give adequate time for these cards to come to room temperature, just like the other reagents that are used during the antibody identification. I will be using 2 pipettes during this test. The first pipette that I use is just a standard pipette, which you should be familiar with. This second pipette allows me to draw up more plasma than what is necessary and dispense a predisposed amount. As you can see, this pipette only allows you to do 12.5, 25, or 50 microliters. Like I previously mentioned, we will need to make a cell suspension for the auto control well. These 2 diluents are used to make that solution. Though they may not be the ones used for the manufacturers, we will be using the 2 plus to make this suspension. As you can see from the bird's eye view, these pumps release a predetermined amount of diluent into the test tube. I will be using 2 pumps of the 0.5 milliliters into a test tube that is properly labeled with the patient's 2 identifiers. After placing 1 milliliter of the 2 plus diluent in the tube, I place 10 microliters of the red blood cells into the properly labeled tube and then mix gently to ensure that there is an even distribution of red blood cells in the tube. This will be used for the patient's auto control well. This is just a quick demonstration of me putting 50 microliters of the red cells from panel 11 into the 11 cell on the IgG card. It is right next to the auto control for the patient. When you are doing the antibody identification, you always begin with the red cells first. You also want to be very careful with your pipetting. You want to ensure that you do not touch the edges of the well, or you may have cross-contamination, which can affect your results. You will also want to ensure that you change your pipette tip in between each well. Lastly, in the auto control well, you will want to place the red cell suspension that you made from the patient into this well. Though it may be difficult to tell, 25 microliters of patient plasma has been added to each of the wells on the IgG cards. To do the plasma, I use the pipette that allows you to do multiple aliquots. It is recommended to change the tip between each IgG card. At this point in time, carefully place your IgG cards in the square portion of the centrifuge. This will allow your IgG cards to incubate for 15 minutes before they are centrifuged. Once your cards have been properly incubated, you can place them in the centrifuge, ensuring that it has been properly balanced. Then you can centrifuge them and wait until they are done. And before I show the results, here is a reminder of how we properly grade gel reactions. As you can see, there is negative, 1, 2, 3, 4, and a mixed field. And just so you can visually see the results, here are the cards 1 through 8. The others will follow shortly. And lastly, there will be the auto control. This will let you know if you're dealing with an autoantibody or an alloantibody. At this point in time, all of your testing is done. It is time to grab the anagram and to place all of the corresponding information from your IgG cards into the proper location on the anagram. The antibody identification is about to begin. Here is the anagram before I place in all of the corresponding information. Like I have previously stated, I have already checked the lot numbers to ensure that I have the correct anagram. I will also be placing 2 patient identifiers to ensure I have the correct patient to the correct antibody. Here is the anagram with all of the corresponding information placed in. We will be using the negative cells to rule out different antibodies. Before we begin, we do need to remember that the rhesus, Duffy, KID, M, N, and S antibodies show dosage. This means that we need to be careful when we are trying to rule out these antigens that have a heterozygous expression of the antigen. First and foremost, I highlighted the groups up in purple which show dosage to have a visual reminder while I'm doing the antibody identification. I used the 3 negative panels to rule out some antibodies. I used a backslash in the screen to do this. If the antibody was positive, I was able to rule it out, except for the ones that showed dosage. If the antibody shows dosage, I looked for the allelic pair. For example, M and N, S and little s. If the allelic pair was heterozygous, I could cross out the positive antibody. However, if it is homozygous, I cannot rule out the antibody as of yet due to the potential of the antibody showing dosage. One trick that I was taught was to use a forward slash on the antibodies that use dosage so that you can keep track of which ones show dosage and you can be prudent with ruling them out. If you look closely at the top, you can see that some of them have been crossed out with an X. If the antibody has been ruled out 3 times in the panels, then it is crossed out with an X. Or if the antibody has been ruled out by a homozygous and a heterozygous antibody, then it has been ruled out with an X as well. Unfortunately, we did not have very many negative panels to work with. Now we need to go forward to the positive panels to see if there are any patterns that we can find. Working with the antibodies that I could not rule out, I have looked for the positive in the positive cells to see if there is a pattern. I have highlighted them for you to see. To have 95% confidence in the antibody that you have detected, you need at least 3 panel cells with the antigen to have reacted with the positive result, and at least 3 panel cells lacking the antigen for your suspected antibody that did not react or in the negative result. Now, as you can see, M fits that pattern perfectly. However, we are still not able to rule out S. Depending on your facility and their SOP, you may need to grab a completely separate panel to rule out that anti-S. Also ensure that you are properly documenting all of your work in time and submitting all of your findings to the proper sources. Thank you for taking your time and watching my video. I would love to hear any feedback below.